Hello, my name is Enikö Sonkoly. I'm a senior lecturer at Karolinska Institute, Stockholm, Sweden. So first of all, I would like to thank the organizers to give me the opportunity to participate in this very exciting course, Genomics for Dermatology. So within this course, I will have 4 talks within the Epigenetics session. This first talk will be about the expanding world of RNAs, MicroRNAs, and beyond. So in this presentation, you will get answers to the following questions. What is epigenetics? What are noncoding RNAs? What are the main types of noncoding RNAs? And why do we have so many of them? So epigenetics means above genetics, if we just translate the word. I think there are different definitions for this word, but in the 1940s, I show you this very early definition. Conrad Weddington gave this definition, the branch of biology which studies the causal interactions between genes and their products which bring the phenotype into being. So very nice. A little bit more modern definition is the following, that epigenetic modifications influence gene expression and alter cellular functions without modifying the genomic sequence. So basically anything that doesn't alter the nucleotide sequence in the DNA, but still alters gene expression and cellular functions can be called epigenetics. This is a quite broad definition. Now there are different areas within epigenetics. One of them is DNA methylation, where methylation of the nucleotides in the DNA can typically suppress gene expression. This is also relevant for skin diseases. However, I think here we are just starting to scratch the surface. There is much more to learn. Histone modifications are also epigenetic modifications which can be relevant for skin diseases. And the third very big area is noncoding RNAs. So these are huge fields, all of them in themselves, and one could talk hours about them. In this talk, I will focus on noncoding RNAs and gene regulation by noncoding RNAs. And to do that, I will go back first to the central dogma of molecular biology. DNA to RNA to protein to function. And this is what we call the flow of information. So what does this mean? This means that every heritable trait of us, from the visible ones like the eye color, hair color, to the invisible ones like biochemical reactions in our cells, are coded in our DNA. And this is something you hear a lot about within this course, I'm sure. However, this code is like a CD, but you have to make music out of it. And how does that work? The DNA is transcribed to messenger RNA, which is translated to proteins. And this is the proteins that are important and that are needed for cellular functions. And the gene is a sequence of DNA that codes for a protein. So according to this central dogma, if this would be the whole truth, you would expect that more complex organisms have a higher number of protein coding genes than the more simple ones. And this is what was actually expected. However, with the Human Genome Project, it turned out that this is not really the case. Because humans and other vertebrates, they have almost the same number of protein coding genes as the worms. Now, of course, one can discuss whether we are more complex than the worms. But I think most of us would agree that we are. And still, we have not so much more protein coding genes, and we have actually less protein coding genes than some protozoa. So that means the number of protein coding genes does not correlate with complexity. Moreover, only 2% of the genomic DNA encodes protein, and almost 80% of the human genome is transcribed. So what is the rest? And protein coding genes, it seems that they may not tell the whole story. The rest, it was considered junk for a very long time. And now, today, we know that much of the non-coding DNA coding for non-coding RNA does actually have a function. So 97 to 98% of transcription results in non-protein coding RNAs in humans. And if you look at the percent of DNA not coding for protein in different species, then we see that this actually correlates much more with complexity than the number of protein coding genes. And also if we look at the percentage of transcription, the non-coding percentage, that also increases with complexity, with 56% in the worms versus 98% in human. So with that said, non-protein coding RNAs constitute the vast majority of the genomic output of complex organisms like us. So based on that, this is the revised central dogma of molecular biology. The core is of course true, but in some cases, and in quite many cases, as you will see, DNA is transcribed into RNA, which we call non-coding RNA, which have regulatory functions. And according to that, I think the definition of gene is also revised, because it's a sequence of DNA that codes for a molecule that has a function, doesn't have to be a protein. And this non-coding RNA was, as I said, for a very long time considered as only junk or transcriptional noise. But now we know that much of these non-coding RNAs, they have actually a function regulating other genes. If we look at the current annotation of the human genome, the non-coding transcripts are actually more in number than the protein coding transcripts. So this is the take-home message from this. Genes are not synonymous to proteins. Much of it is non-coding. So if you do your GWAS studies, if you look your transcriptomic studies, you do not necessarily need to exclude these transcripts right away. So what are these then? This is the tree of RNA types. This is a bit arbitrary. There are coding ones and non-coding ones, and we know that these categories are a little bit overlapping. The coding is something we don't talk about today, but from the non-coding ones, there are the housekeeping and the regulatory, and even we know today some of the housekeeping RNAs can be regulatory. And within the regulatory RNA category, there are the short and the long ones. This is just an arbitrary categorization. The short ones are shorter than 200 nucleotides, and the long ones are longer. And within the short RNA category, microRNAs or miRNAs are the best studied group of non-coding RNAs. So I would like to say here some words about the long non-coding RNAs. Long non-coding RNAs, as I showed you, the definition is dependent on the size. They are longer than 200 nucleotides, as opposed to the short ones that are all shorter. But other than that, they do not have very much in common. This is a very heterogeneous, very diverse class of RNAs. What is common in them, they have little or no open reading frames, so they do not have the capacity to code for proteins, and they have a low sequence conservation typically. And this is also in contrast to protein coding genes, which are usually conserved by sequence. However, the structure can be conserved more than the sequence. Many of these long non-coding RNAs have a low abundance compared to messenger RNAs. And from some long non-coding RNAs, based on how they function, we see that only a few copies per cell can exert a function. But this can make it difficult to detect them and also to study them. Many long non-coding RNAs have highly tissue or developmental stage specific expression. And here are some examples for that. So long non-coding RNAs are typically, on average, even not all of them, but on average, more tissue, cell type, and developmental stage specific in their expression if we compare them to messenger RNAs. And these long non-coding RNAs are probably function by defining cell identity, by regulating stemness, or cell differentiation. So some examples from neural stem cells, skin stem cells, and also muscle stem cells are seen here. Of course, since this is the dermatology course we are all interested in the skin, we see two examples here from the skin, Tinker and Anker. These two long non-coding RNAs are expressed in the skin, and already their function is known. So I will come back to them. So how do long non-coding RNAs work? As I said, they are a very heterogeneous group of RNAs. They can be located in the nucleus or in the cytoplasm, and they work a little bit differently, the nuclear and the cytoplasmic ones, or they can be localized into both the nucleus and the cytoplasm. So nuclear long non-coding RNAs, they can also have diverse functions. They can have an enhancer function. They can also bind to chromatin modifying complexes. One example for this is the long non-coding RNA, which is called Exist. So all of us know that in females, the X chromosome is inactivated. One of the two X chromosomes is inactivated in the cells. But what regulates this X chromosome inactivation has been unknown for a very, very long time. And one factor which is essential for the inactivation of the X chromosome is actually a long non-coding RNA like this, which is called Exist. So it's an interesting mechanism. This long non-coding RNA is coating the whole inactivated X chromosome. Then they can also be transcriptional regulators. They can recruit transcription factors, for example, to the site of transcription. They can also mediate interaction between different chromosomes, and they can also regulate splicing. So these are some examples for mechanisms what long non-coding RNAs can do in the nucleus. And for some of these mechanisms, we don't need more than a few copies of long non-coding RNAs for these functions. And in the cytoplasm, they can also have different functions. They can regulate the stability of messenger RNAs, for example. They can also regulate mRNA translation. Interestingly, some of the long non-coding RNAs are circular, and this was a very interesting discovery. When it was discovered, it was thought that our RNAs are all linear in the cell, but some are circular, and these are called circular RNAs. These are a subclass of long non-coding RNAs, and some of them at least, can function as microRNA competitors. So they can have a lot of binding sites for the same microRNA, and by sponging them, they can inhibit the microRNA they can prevent the microRNA from binding to their targets. So this is a quite interesting mechanism, but probably not very common in biology. And then last but not least, some long non-coding RNAs can code actually for small peptides. And you may think that this is a contradiction, because the very definition of non-coding RNAs is that they do not code for proteins. But these are not real long proteins, but they are very, very short, small peptides, also called micropeptides, from very short open reading frames. And this can be the case sometimes with some long non-coding RNAs. So again, the skin, because this is the organ that we are all specifically interested in. Here are some examples for long non-coding RNAs in the skin. Actually, I think here, again, we are just scratching the surface. There is a lot more to discover. Many more long non-coding RNAs for which the function is completely unknown, both in the skin and in other organs. So one example is PRINCE. This was the first long non-coding RNA ever associated to any skin disease. And this is a long non-coding RNA which we found in psoriasis, in the non-lesional epidermis of psoriasis patients. And this long non-coding RNA turned out to be regulated by stress. So this was a very early example, and after that many others have been, or some others have been shown in the skin. I show you here also two other examples of long non-coding RNAs, Anker and Tinker. As I said, some long non-coding RNAs define cell identity, stemness, differentiation. This is like a pair of long non-coding RNAs with opposite functions. Anker is expressed in more like in the basal layer of the epidermis and represses differentiation. Tinker, on the other hand, is expressed in the differentiated keratinocytes and promotes differentiation. So I am sure that there are many more to come in the coming years. So again, coming back to the tree of RNA types, these were the long RNAs, the long non-coding RNAs, longer than 200 nucleotides with very diverse functions. And then I would like to say a few words about microRNAs as well. So microRNAs are the best studied group of RNAs and of regulatory non-coding RNAs. And they were discovered actually 1993, the very first one. But then we didn't know that it was a microRNA. A gene was found in the worms, in the Caenorhabditis elegans. It's a model animal. And there was a development of phenotype. The researchers were looking for the mutation, and it was in this gene. However, the corresponding protein was never found. And then the conclusion was this is a non-protein coding RNA. And later, in the Victor Ambrose group, it was also discovered that this RNA is regulating a protein coding gene. However, this was at that time considered as a peculiarity in the worms, and which does not have any significance for other animals or for human. So the field was completely dead for 7 years, nothing happened until another microRNA, LET7, and another target was found year 2000. And LET7 exists, it's a well-conserved microRNA family, which exists in many species. And then the field just exploded. 1 year later, hundreds of short RNAs were found in plants, worms, flies, and vertebrates. Many of them were highly conserved and developmentally regulated. And the official term microRNA was introduced in 2001. So there are now a high amount of publications on microRNAs in different organs. So some of the microRNAs are highly conserved. As I said, this is one example, the LET7, which is identical from flies to worms to humans. So it means that this microRNA already existed hundreds of millions of years ago, long before the dinosaurs, in the last common ancestor of flies and worms and human. Very interesting. And also there are many, many other microRNAs that are conserved. miR1 differs by 1 nucleotide in Drosophila and Homo sapiens. miR203, a skin microRNA, differs only by 1 nucleotide between zebrafish and human. So we have many microRNAs, but if we look at the number of microRNAs, it is increasing with developmental complexity, just like non-coding RNAs in general. As we can see, so humans have more than 2,500 microRNAs. And it seems that microRNAs during evolution, they were continuously acquired. So why do we need so many? As you see here, shown in another way, so almost in every branching point in the evolutionary tree, at least one new microRNA gene appeared and not so many disappeared. And this is actually in contrast to transcription factors, which are also gene regulators. Very few, if any, transcription factor families have evolved during evolution, while the microRNAs, the number of microRNAs has been really increased. So I will tell you more about why we need so many microRNAs in the coming presentations. So I would just like to summarize this talk. In our transcriptome, the vast majority is non-coding, does not code for protein. But these non-coding transcripts are usually regulatory. They are not just transcriptional noise, not all of them at least. Non-coding RNAs are a very heterogeneous group of RNAs which regulate the expression of other genes. and the number of non-coding RNAs increases with developmental complexity. Long non-coding RNAs are a heterogeneous group of RNAs longer than 200 nucleotides. They regulate also gene expression in many different ways. And microRNAs are the best characterized group of non-coding RNAs, and I will talk more about them in the coming presentations. Thank you for your attention.